Hello, everyone, and welcome to today's webinar, Facilitating High-Throughput, Large Cohort Proteomic Analyses Using an Integrated Software Solution. I'm Christina Joel of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize this Q&A box and we will assist you directly. I would now like to welcome our speakers, David Horn, Senior Product Manager, Thermo Fisher Scientific, and Kevin Schauer, Senior Product Manager, Thermo Fisher Scientific. For complete biographies on our speakers, please visit the Biography tab at the left of your screen. David, welcome. You may now begin your presentation.
Thank you very much for joining this webinar. Today, we are going to give you an overview of the Thermo Scientific Proteome Discoverer software and the Thermo Scientific Ardia Platform for proteomics data analysis. We will show you throughout this presentation how these two platforms work together to analyze complex proteomics datasets. I am David Horn, Product Manager of Proteome Discoverer, and I will lead the first part of this presentation. One of the major challenges for the analysis of data from Thermo Scientific mass spectrometers is the sheer diversity of acquisition methods used to support various proteomics, protein characterization, and structural biology workflows. Acquisition methods may include multiple mass analyzers, multiple methods of fragmentation, multiple levels of fragmentation, and a diversity of quantification methods that challenge downstream data analysis tools. There are quite a few software platforms that can support any one of these acquisition methods, but there is only a single software package that supports all of them. The Thermo Scientific Proteome Discoverer software is our solution for supporting these various proteomics workflows. PD, as we call it, is not simply a search engine, but instead it is a platform performing proteomics and structural biology data analysis and interpretation. The software is based on a node-based workflow engine, making it relatively easy to deploy new technology into the platform. We can, of course, perform standard peptide ID and quantification using algorithms developed internally, as well as with third-party partners. We can also perform post-translational modification analysis such as phosphopeptide ID and provide modification site localization confidence. Third, we support cross-link peptide identification workflows using the X-LinkX nodes for Proteome Discoverer in partnership with Utrecht University. Finally, we also developed the ProSightPC software in collaboration with Proteinaceous for top-down proteoform identification. Let's delve into more details about the features in the Proteome Discoverer software. First, PD supports many of the most commonly used quantification strategies. We have invested a substantial amount of effort in implementing a full TMT workflow that includes MS2 and MS3 based approaches with correction factors, scaling and normalization, and specialized visualization for results interpretation. More recently, we have partnered with MSA to implement chimeras in the Discoverer platform to support data-independent analysis, improved data-dependent analysis, and to some degree parallel reaction monitoring. Proteome Discoverer also supports MS1-based label-free quantification, as well as isotopically labeled quantification methods such as SILAC or dimethyl labeling. Secondly, the Proteome Discoverer software is based on a customizable node-based workflow system. While we have several dozen default workflows to choose from, workflows can be further customized to include multiple search engines or more complex search strategies to maximize the amount of information that can be interpreted from any dataset. This workflow-based system also includes filters that can send through spectra for a given mass analyzer, fragmentation method, or MS level to use with a different search approach than for other branches in the workflow. This node-based approach is extensible and allows us to partner with third parties to include new search methods in the platform. The search algorithms that we co-develop are shown in the list here, but there are other nodes developed by third parties that can be downloaded directly from their websites. The Proteome Discoverer software also includes several AI/ML methods. These include the AI-based algorithm inFIRS for spectral prediction, as can be seen in the plot to the upper right. We can predict theoretical MSMS spectra on the fly and compare to the experimental spectrum for visual validation. inFIRS is also included in a peptide identification workflow called inFIRS for scoring, and is also included in the Chimeras algorithm that I will describe later. We also include tools for statistical analysis and visualization, and of course use machine learning tools to calculate the false discovery rate at the PSM, DIA precursor, peptide group, protein, and protein group levels. The Proteome Discoverer software includes tools for study management that allows assignment of study factors to quantification measurements. This feature has multiple purposes, including setting up replicates for quantification and ratio calculations, as well as visualizing results by condition in the tables and charts rather than by file or quant channel. Study management only requires a small upfront time investment to set up study factors for analysis, with ample returns on that investment in helping make scientific conclusions. The Proteome Discoverer software provides powerful tools for results visualization. Each of these plots are linked back to the tables that were used to source data, letting users highlight, select, and analyze related proteins or peptides. Such views include volcano plots, heat maps, PCA plots, scatter plots, bar charts, Venn diagrams, chromatograms, and spectra. We also include hierarchical tables that are easily filtered to help you find the proteins or peptides of most interest. Finally, we provide tools for bioinformatic analysis using an annotation service that we host in the cloud. When the protein annotation node is included in a workflow, the protein table in the results is annotated with several ontologies, including gene ontology terms, pathways, protein families, chromosomes, gene symbols, and more, with links to pathway diagrams. We also provide enrichment analysis, which returns gene ontology or pathway terms that are significantly over or underrepresented in a selected set of proteins in a dataset. In summary, Proteome Discoverer provides a foundation that supports our advanced acquisition methods from Thermo Scientific mass spectrometers and powerful tools for visualization that help you translate raw data to biological discovery. In the next few slides, I'll further highlight our collaboration with MSA to incorporate their inFIRS and Chimeras tools into the Proteome Discoverer framework. inFIRS is a deep learning-based algorithm for prediction of peptide MSMS spectra. The majority, but not all, of the input data for the inFIRS algorithm came from the annotated peptide MSMS spectra acquired for the Proteome Tools project led by the Küster Group in Munich. In this project, 1.4 million peptides were synthesized and acquired using multiple fragmentation energies and fragmentation methods, resulting in over 100 million spectra with sequence-specific properties. This data set is ideal for deep learning and are the basis for why inFIRS predicts MSMS spectra so accurately. MS8 is a company spun out of Bernhard Küster's group and has continued to refine and develop this early work into inFIRS and Chimeras. Intelligent search algorithm that works with various acquisition methods such as DIA, DDA, wide window acquisition, and PRM. It is built upon the inFIRS spectral prediction algorithm and goes multiple steps further to deconvolute complex MSMS spectra to produce peptide IDs and quantification values in the case of DIA and PRM acquisition. Chimeras does require a specialized computing environment, given that it is built on AI tools, and is thus either deployed on the cloud or, as you will see later, on one of our Ardia Platform servers. Chimeras performs quantification in a unique manner. The Chimeras algorithm itself produces a relative quantitative value for any given peptide in an MSMS spectrum when it performs deconvolution. For DIA or PRM, Chimeras calculates the quantitative values for all PSMs in consecutive DI cycles across the elution profile of a peptide. The user can choose whether Chimeras returns the apex abundance or the full peak area for any given peptide for use in peptide and protein quantification. This is thus not a targeted approach, but instead the peptide is identified independently in each spectrum across the elution profile. Proteome Discoverer software treats these quantitative values similarly for DIA as it does for TMT and LFQ, rolling up the DI precursors to peptide and protein quantitative values. The Proteome Discoverer software version 3.2 is soon to release at the time of this live presentation. This release includes an updated protein group FDR calculation, support for the new TMT 32 and 35-plex reagents, publishing the protein, peptide, and PSM counts to the Ardia platform when PD uploads results, as I will show later, and many other improvements not listed in detail here. Chimeras also has been further improved. The search is considerably faster than in previous versions. It supports new PTMs and includes a new PTM site localization algorithm for DI data. It supports a dramatic increase in the upper dataset size limit when running full experiment in one go, and semi-specific enzymatic cleavage for datasets where you would expect to see such peptides. While I hope I've shown that the Proteome Discoverer software is a powerful and flexible platform for proteomics applications, there are some limitations to standalone PD when dataset complexity increases. PD is designated for installation on a local PC with the subsequent limitations of storage space and computing capacity. As datasets are readily eclipsing 1 terabyte in size, hard drive space is increasingly becoming a limitation for local software. PD also requires data to be manually transferred from the instrument PC to the data analysis PC prior to analysis, and we usually wait for the full dataset to be acquired before running the analysis. However, for very large datasets, it is advantageous to accumulate results as they are acquired rather than waiting for the acquisition to finish for all files. Finally, the cloud version of Chimeras ultimately has limited capacity and is generally insufficient to analyze the largest Orbitrap Astral datasets. A local, higher capacity version of Chimeras will be necessary to support data from multiple instruments as well. How might we address these issues? Before I go into more detail, I do want to mention that we released an important update to Proteome Discoverer 3.1 earlier this year that is instrumental to support larger datasets. We added a single parameter to the Chimeras node that dramatically reduces the PD analysis time for large DIA datasets from the Orbitrap Astral Mass Spectrometer, as well as our other MS systems. This, of course, is a welcome improvement for throughput, but it does not solve the other problems listed on the previous slide. However, if you are a Proteome Discoverer 3.1 user, this feature is included in the latest service pack, and I encourage you to download and install it. For the other issues I had mentioned, the Thermo Scientific Ardia Platform can help. This platform is software for scientists running chromatography and mass spectrometry experiments, and it is built on the latest software architecture and technology. Before giving a brief overview of the platform, I'd like to mention why we built it. We built Ardia because we listened to what the scientific community was saying, and this is what we heard. We have a lot of data. Our data is distributed across multiple labs, and we need a better way to manage it. This is a common sight we see in labs with lots of portable hard drives and multiple copies of data. This can make retrieving data from historical studies extremely challenging and very time-consuming. People also need an easier way to share data, methods, and results with colleagues, collaborators, and clients. Improvements in lab productivity is always top of mind, so more automation, faster processing, and better utilization of instruments is necessary. And we're now living in the AI era, so people want to leverage their historical data, look for predictive patterns, and gain deeper insights out of their studies. The Ardia platform is a centralized hub that connects your instruments and application software. Whether you are running Chromeleon or Excalibur, data is stored on Ardia, which provides easy access to anyone that has an Ardia account with privileges to view that data. You can get instrument status for your whole fleet from anywhere you have access to the Ardia logon. We are also connecting our existing software applications to provide them with enhanced capabilities that they don't have natively. These include user management, audit trails, and results versioning, automation and improved processing performance, an advanced report builder, and tools to gain deeper insights into the connected data. In this presentation, I will only mention one core Ardia tool. In the Ardia Instruments application, each tile in the interface represents an instrument or system. This example includes GCMS systems, standalone LCs, and an LCMS system. Any instrument controlled by Excalibur or Chameleon can be connected to Ardia. The tiles display acquisition status, current sample progress, remaining samples in the injection list, and the expected batch completion time. Additionally, a real-time mini plot of the chromatogram is shown to allow visual verification of peak presence and sample quality. Clicking on a tile provides more detailed information. A built-in scheduler is also included, allowing you to book time on instruments. This functionality extends beyond connected instruments, enabling scheduling for any equipment in your lab. This application can be very useful for larger proteomics datasets where multiple instruments are simultaneously acquiring data. This gives a single overview of instrument status, while the Ardia system provides common storage for acquired data from each instrument. I'm not going to go into further detail about the features of the core Ardia platform, but instead we'll highlight those features of Ardia that provide benefit in combination with Proteome Discoverer for larger proteomics datasets. For more information about the Ardia platform, please go to thermofisher.com and search for Ardia Platform. I also invite you to view the recent webinar from Mark Sanders, who provides a terrific overview of the Ardia platform in combination with several of our software applications. Currently, we are offering the Ardia platform on an on-premise server, providing you with 3 options based on your specific application and storage needs. Our most powerful option is the Advanced Tower server, which is the best choice for proteomics applications. This server not only offers the on-premise Chimeras intelligent search algorithm, but also provides an impressive amount of storage. Proteome Discoverer can indeed connect to the Ardia storage system, and thus the upper limit of dataset size is greatly increased using Ardia compared to a standalone PC where all raw data and PD results must be stored. We also provide a powerful reference data processing PC that we have tested thoroughly with Proteome Discoverer and Ardia to guarantee performance of the automated workflow that I will describe later. When you place the Ardia Advanced Tower server in your laboratory, any licensed Proteome Discoverer 3.1 or 3.2 installation that is connected to that server has access to the local Chimeras algorithm. The Chimera service on the Ardia server has substantially higher throughput than any of the cloud versions we provide, and is much more capable of keeping up with our highest throughput instrumentation. Further, the use of Chimeras on the Ardia server keeps all data within your firewall and thus can be used in more lockdown IT security environments. You can ensure that you are using the local version of Chimeras by using the Chimeras on Ardia server node as shown here. The cloud version of the node is simply called Chimeras and requires a cloud account to connect to the service. A second benefit that Ardia provides for large proteomics datasets is automated processing. If an instrument PC with Excalibur and a data analysis PC with Proteome Discoverer are connected to the same Ardia Advanced Tower server, Proteome Discoverer workflows can be selected within Excalibur for automatic post-processing. In this case, once the run is started, data is acquired on the instrument control PC and then automatically transferred to the Ardia server when acquisition is complete. If the file is designated for automated processing, the Ardia server automatically initiates a Proteome Discoverer analysis on the data analysis PC designated for automated processing. If the analysis includes Chimeras, Proteome Discoverer will further trigger the run on the Ardia server and wait for the results to come back. When the Proteome Discoverer analysis for the file is complete, the Proteome Discoverer result is then moved to the Ardia server. Thus, acquisition and processing will happen simultaneously, offering a significant time-saving advantage. Once all files for a proteomics experiment are acquired and searched individually, then you import the individual result files into Proteome Discoverer study, apply the study factors and set up statistics, and then generate a consensus report containing the combined results. The resulting file can then be published to the Ardia server, keeping all data and results together and allowing others to access them through an Ardia login. This makes it a comprehensive solution for high-throughput proteomics. The capability to register a PD installation on a data analysis PC for automation can be seen here. In the administration tools, there's an automated processing configuration screen. When the checkbox at the top of the screen is checked, Proteome Discoverer informs the Ardia server that it is ready to receive jobs for processing. The PC that receives these automated jobs needs to be powerful enough to keep up with our latest generation of instruments, and thus the processing PC mentioned a few slides back is highly recommended. The user can also select which workflows are available for automated processing from within Proteome Discoverer. Only those analyses published using the method shown here will be made available for use with automated processing. It is important to note that the workflows available for automated processing are not limited to DIA workflows using chimeras, but other workflows using different search engines and quantification approaches can also be selected. When the workflow is saved for automated processing, the Ardia server is notified and the workflow will be added to the list that you can see in Excalibur. When Excalibur is connected to the same Ardia server as the installation of Proteome Discoverer that is registered for automation, a checkbox called Ardia Platform Processing with Proteome Discoverer software is shown above the Excalibur sequence. After activating this option, 3 extra columns appear that allows users to select the analysis method, the PDQuan method, and the target results path for automated analysis. After each file is acquired, the file will be uploaded automatically to the Ardia server in the path chosen by the user, which will subsequently trigger a Proteome Discoverer analysis on a connected data analysis PC. After the PD analysis has completed, the result file will be saved to the result path folder as selected by the user in the sequence. The advantages of automated processing are shown here. In a traditional PD analysis, the full dataset is collected and then manually transferred to a processing PC for the search. Then the full analysis of all data is triggered, and finally a result is produced. With the Ardia Platform automation, data is collected and searched in parallel, allowing up to 20 files to be searched simultaneously without performance reduction. Furthermore, all raw data files and results are automatically transferred to Ardia storage, eliminating the risk of the instrument PC or the data analysis PC filling up. This results in significant time savings while also reducing the stress of having to manually manage file transfer between systems. Further, you can look at the results of any data file during the accumulation of experimental data to ensure that the results are as expected. Ultimately, the time from acquisition to result file is substantially reduced due to the automation while keeping all your raw data and results in centralized storage. One final new feature we would like to highlight is the annotation of Proteome Discoverer results in the Ardia platform. Here I am showing a screenshot of our Data Explorer Ardia application. When results from Proteome Discoverer are uploaded during automation or are manually published from a PD instance, the protein, peptide, PSM, and MSMS spectra counts are annotated in the file list. We will only annotate files for results that are FDR controlled. These numbers can give you insight into the complexity of each sample and can help you catch issues with sample preparation or other aspects of the system if the numbers change substantially as data are being acquired. Finally, the Ardia platform is not limited to a single instrument or single application client. Although only one Proteome Discoverer data analysis PC can be designated for automated processing with the given Ardia server, processing is usually fast enough to keep up with 2 Orbitrap Astral instruments or up to 10 hybrid or tribrid instruments. Further, you can connect many more client PCs to the Ardia server to access the storage system. We do not limit the number of Proteome Discoverer clients that can connect to the Ardia platform to access raw files and publish results, and you can also connect other software applications such as Biopharma Finder or Compound Discoverer to the same Ardia server. Although I've been focusing on our Proteome Discoverer software application, we are committed to an open platform. If you choose to process data with non-Thermo Scientific software, we enable to use the software tools of your choice through the Ardia Data Sync application. This small program runs on a processing PC where the third-party software is installed. It provides login capability to Ardia and allows you to designate folders on the Ardia server to sync to the local computer. As soon as data appears in the designated folder on Ardia, it is mirrored to the processing PC on a file-by-file basis. Thus, you can have all your files automatically transferred to your desktop PC immediately post-acquisition by uploading to Ardia using Excalibur and then downloading using Ardia Data Sync. That completes my portion of the presentation, and now I'm going to pass the presentation over to Kevin.
Hello everyone, thanks so much for joining. Today I'm going to talk you through a study that we performed with a collaborator where we're looking at proteomic changes that occur during the storage of blood cells from non-domesticated felids, sometimes referred to as big cats. Just to give you a little bit of background on this study, we worked with a collaborator who's a veterinarian at the Brookfield Zoo in Chicago, Illinois. This collaborator is interested in potentially creating a blood bank that could store blood from a variety of different species and could use this blood for life-saving treatments both at the Brookfield Zoo as well as across other zoos in the country. However, it's well known that in domestic cats, the storage of blood is difficult. Unlike storing the blood from other species, such as humans, domestic cat blood degrades relatively quickly when stored. This makes it difficult to maintain large blood banks and to store the blood for long periods of time. Our collaborator was interested, however, to see if the similar trends were present in these larger cat species, or if it may be possible to store the blood from these species long term. To perform the study, blood from 134 different individual cats spanning 18 different species were collected from zoos across the United States. All the blood was drawn either via voluntary behaviors, such as the one shown in the picture on the bottom of the slide, or as part of previously scheduled procedures that would allow for collection of blood for the study without any additional stress to the animals. Once the blood was collected, it was shipped to our collaborator, and the blood was used for a variety of different studies, including cross-matching, gene sequencing, and the proteomic analysis, which I'll be discussing here. In an effort to better understand how resilient blood from these species is to long-term storage, some of the collected blood was stored in a blood storage refrigerator, and aliquots were taken after 0, 7, 14, and 28 days of storage. The blood collected after the storage period was then centrifuged, and the resulting cell pellet was washed several times in a saline solution and then stored at -80 degrees C for proteomic analysis. Overall, the samples from the 134 individuals resulted in approximately 540 total samples for proteomic analysis. Each sample was injected in duplicate, resulting in over 1,100 total injections on the mass spectrometer. This excludes blanks and QCs that were also part of the sequence. The distribution of the individual across the species studied here is shown in the table at the right. Once all of the samples were collected, we started with preparation for the proteomic analysis. Due to the large number of samples that were needed to be processed, we really wanted to find a protocol that would require little hands-on intervention. The Accelerome sample preparation platform, which is specialized for the preparation of proteomic analysis, was well suited for this. Briefly, a lysis buffer was added to the cell pellets, and after a brief incubation, the protein concentration for each sample was determined using a nanodrop spectrophotometer. Once the protein concentration was determined, 75 micrograms of each sample was added to the Accelerome sample preparation plate, and the Accelerome platform performed the reduction, alkylation, digestion, cleanup, and peptide concentration measurements. The resulting plate was then dried down in a speed-back vacuum concentrator, and the dried pellet was stored in the freezer until analysis. Analysis was performed on an Orbitrap astral mass spectrometer coupled to a Vanquish Neo UHPLC. The Vanquish Neo was operated at capillary flow rates ranging from 2 to 2.5 microliters a minute. The total gradient used was about 13 minutes long, and this excluded the loading and equilibration phases, which took approximately 3 minutes on average. This allowed for the analysis of approximately 100 samples per day. 200 nanograms of peptides were loaded on column for each run, and we did use a trap-elute setup here, and we utilized the zebra-ROS function, which is part of the Vanquish Neo, to reduce carryover between analysis. A short sawtooth gradient lasting approximately 30 seconds was also added to the end of each of the runs to add in as additional cleaning step for the analytical column. The Orbitrap Astral Mass Spectrometer was operated in data independent acquisition, or DIA mode, using the parameters shown in the right column. Briefly, the precursor mass range was set to 390 to 980 m/z with an isolation window of 3 Thomson. This resulted in approximately 200 scan events per cycle. MS1 scans were performed every 0.6 seconds in the Orbitrap with a resolution of 240K. The Excalibur instrument PC that was connected to the mass spectrometer was also connected to the Ardia platform, as is described by Dave in the previous section. This connection simplified data processing and also made it easy to monitor progression of the analysis over the nearly 2 weeks that the data was acquired for the sample set. Prior to the collection of the main data set, we did run a subset of the samples containing just the 0 and 28-day storage time points. All of these derived from a single individual from each species. We then analyzed these using Proteome Discoverer, and this pilot study, which took us less than 24 hours, was used to generate an inclusion file that was then used for the searching of the rest of the full data set in Proteome Discoverer. Using automation between Excalibur and Proteome Discoverer that was enabled by the Ardia platform, data from each run was searched using the Chimeras algorithm immediately after data collection of each run completed. So the run would finish, be uploaded to the Ardia platform, and then immediately analysis would start with Proteome Discoverer and Chimeras. As the Ardia platform is capable of performing up 20 Chimeras searches in parallel without slowing down or reducing performance, the processing steps were able to keep up with the data acquisition, allowing for all the searches of the individual injections to be completed within hours of the final acquisition completing. These individual processing results were then combined into a single multi-consensus result. As Dave mentioned in his presentation, when PD results are uploaded to Ardia as part of this automated analysis pipeline, it's possible to display some of the summary statistics for each result in the Data Explorer. We found this for this large data set to be really useful, as we could just check in daily or so to check and make sure that our identifications were in the range that we were expecting. This was particularly nice when the Gila vial that we were using as a QC ran out in the middle of a long weekend. We were able to see an obvious drop-off in the protein identifications after row 26 here, but we are able to see that our data from the actual samples continued with the number of identifications as expected, clearly indicating that it was the HeLa that was the problem, not the instrument or the other samples. Before diving into the results, I wanted to mention a couple of challenges in processing this data set. First of all, as one of the main purposes of this study was to compare across species to see if different species were more resilient to blood storage than others, we wanted to use the same protein database for all of the samples. This would just allow for a more simple comparison. This, along with the fact that genomic or proteomic data is lacking for many of the species studied here, meant that we had to use a large database for these analyses. We chose to use all UniProt KB entries belonging to the suborder Feliformia, including all of the subspecies. This resulted in a FASTA containing nearly 300,000 sequences. In addition, the database contained a significant amount of redundancy, adding additional challenges to the searches. Despite these challenges, we were able to get exciting results. In total, the study contained over 88 million MS2 spectra, which resulted in nearly 15 million peptide spectral matches, or PSMs. This mapped to identification of about 53,000 peptide groups and over 8,000 protein groups. This slide provides some more detail on the identifications across the different species. Overall, we identified approximately 11,000 to 12,000 peptide groups for each individual injection across the different species, and these map to approximately 1,500 protein groups per species. As a note, there's likely some redundancy in these protein counts simply because of the redundancy in the database, so these numbers may be a slight overestimation of the total proteins identified. Found these numbers to be specifically impressive based off of the large dynamic range of the sample sets. So here we looked at the abundance of the proteins for just the cheetah samples, simply to make things a little bit more simple to look at. The protein abundance identified across the different samples showed a very impressive dynamic range that the Orbitrap Astral was able to handle. In fact, we identified proteins with over 7 orders of magnitude difference in abundance between the highest abundance and the lowest abundance. You can see that hemoglobin and albumin, not surprisingly, were of the highest abundance, and they were more than an order of magnitude higher than essentially all of the other proteins identified. In most cases, they were more than a couple orders of magnitude higher. Regardless, we were able to identify thousands of other proteins with much lower abundance, and they were across many of the samples. We also looked at the consistency of the data across the study. As you can see from this plot of the non-normalized sum peptide abundance, the data was incredibly consistent across the nearly 14 days of acquisition. There was one slight drop-off between injections 200 and 300, at which time we identified an issue with the trap column, which was replaced. After replacing the trap column, the abundance was immediately returning to the starting level and maintained there for the duration of data collection. Now, since we looked at some of the overall quality metrics for the data set, let's dive into the bit of the biology. We wanted to see how the different samples compared between species overall. Here we show a principal component analysis on the left side that highlights the differences between species, and the similarity between time points. Each color represents a different species, and there are 4 of each of these colors representing the 4 different storage time points. You can see here a clear differentiation between species, but a close clustering of the different time points. Impressively, the clustering of the different species very closely matches the relatedness of the different species, as shown in the genetic tree on the right. If we look in the blue box, you can see that there's clustering of the closely related bobcat and lynx samples, and in the red box, you can see the closely related lion, amber leopard, jaguar, tiger, snow, and clouded leopard samples. Overall, the differences between species accounts for the very large percentage of the variance across the entire data set. We next started looking at changes in both the peptide and protein abundance across the different storage time points. I'll simplify things here by only showing you the data from the approximately 156 cheetah samples that were analyzed. Here, I'm showing the volcano plots to illustrate the peptide abundance at the different time points relative to 0 day. To be honest, these results were quite surprising to me and not what I was expecting. I was expecting to see decreases in peptide abundance that could be indicative of chemical degradation as storage time increased, as I expected that most of this blood would be relatively biologically inert during the storage. However, that's not at all what we saw. In fact, we saw that there were a number of peptides that were increasing abundance, and they become more increased in abundance as storage duration increased. This suggested that many of the changes are not actually due to chemical degradation at all, but there's actually quite a bit of biological processes that are proceeding even during the storage. After seeing this, we also looked into the protein level, which I again found surprising. Again, I expected to see few changes in protein abundance, with most of the changes being decreases due to chemical degradation during storage. Again, this trend was reversed, and at the protein level, with longer storage time showing significant upregulation of proteins relative to day 0. So we then took a look at some of these proteins that were indeed increasing in abundance, and we looked at these with the collaborator to see if there's anything of biological interest. Indeed, there were. There are a number of different classes of proteins that were really intriguing to our collaborator. First of all, there were a number of different clotting factors that showed increased during storage, suggesting that there's active modulation of both pro and anticoagulant factors, even though the blood was stored in an anticoagulant. Paraoxidase and serotransferrin upregulation also suggests that cells may be attempting to deal with oxidative damage and the degradation that could result from this late time storage. This is something we plan to look into in the future by looking at changes in oxidation across time points. Further, apolipoprotein upregulation may indicate that the red blood cells are switching to metabolic pathways other than glucose, as that glucose is likely becoming depleted as blood was being stored. Finally, we saw that there were complement proteins that showed upregulation, and this could signal an increase in the innate immune system via the white blood cells that are stored in the blood unit. In conclusion, we found here that the Proteome Discoverer software powered by the Ardia Platform allows for the analysis and management of large cohort, high-throughput analyses, even for these bioinformatically challenging multi-species studies like the one we discussed here. The automation of acquisition and data processing via Excalibur, Proteome Discoverer, and Ardia simplified data management, and it really reduced the processing time for these large studies. The Vanquish Neo and the Orbitrap Astral instrument combination is really an ideal platform for these types of high throughput proteomic studies, allowing you to get high throughput without sacrificing depth of coverage. The study really exemplifies the power of this platform due to the high dynamic range of the samples and the fact that we were able to see over 7 orders of magnitude differences across proteins identified. Finally, the Accelerome platform really simplified sample preparation, allowing for these large number of samples to be prepared with minimal user intervention. With that, I'd like to thank our collaborator, Dr. Lily Parkinson of the Brookfield Zoo, for allowing us to work with this very exciting sample set. I'd also like to thank Amir Hakimi and Kevin Yang for their invaluable help with the sample preparation, instrument setup, and much more. They were really kind of the eyes, ears, and hands of this operation as I just directed from afar. We'd also like to thank all of the contributing AZA institutions, or the Association of Zoo and Aquarium Institutions, that contribute samples to the study, and finally the University of Georgia for funding. If you're interested in getting any more information on this data set, I presented a poster at ASMS this past June that can be found at the website here, thermofisher.com/ASMS. In addition, we published an application note about a year ago that was a preliminary study on the same data set that used a subset of the samples and the Orbitrap Explorers platform. So feel free to check that out in this application note as well. With that, I'd like to thank you for your attention, and now I'd be happy to move to any questions you may have.
Thank you, David and Kevin, for those excellent presentations. It is now time for our live Q&A portion of our webinar. Now, we have lots of questions that have already come in, but if you haven't submitted any yet, please do so now. Simply type them into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. So, David, we already have so many questions. I'm going to dive in. Let's just start with this one. In which version of PD is the DIA analysis implemented?
Thanks for the question. I actually had a slide that showed the full history of the Proteome Discoverer software that goes way back 15 years that talks about each of the major features. Fortunately, I removed it, but I'll talk through it as best I can. So maybe the way to discuss this is just the history of our timeline of our collaboration with MSAID and getting these AI-based kind of tools into Proteome Discoverer. So our first algorithm that we did in collaboration with MSAID is an algorithm called inference rescoring, and that was introduced in PD 2.5. And that algorithm, you'd use it for things like immunopeptidomics, TMT, and what it'll do is it'll predict the spectrum of that identified peptide using the AI-based tool inference, and then we'll correlate the predicted spectra and then measure spectra, and then that spectral angle and some other figure of merits would boost its confidence of a result if it matched correctly. And then in PD 3.0, we released the first version of Chimeras, which was for DDA analysis. So it did not yet support DIA analysis, and it was in PD 3.1 where Chimeras supports DIA analysis. And then in PD 3.2, as I showed in the slide earlier in the presentation, we were able to improve its performance, of course, in partnership with MSA to make it faster, support new modifications, and so forth. So Proteome Discoverer 3.1, which is the most recent release, is the one that supports DIA.
Thank you, David. And I'm getting quite a few questions in regards to interest here of wanting to try Chimeras. Is it possible to try it out?
Yes. So you can, first of all, you can try Proteome Discover demo. So we have the site thermo.flexnetoperations.com where you can download the latest versions of Proteome Discover to demo the whole software. Chimeras is a special case that's not automatically working with that demo because the Chimeras service, unless you have it on the Ardia server, will primarily be in the cloud. And the cloud version of Chimeras has its own kind of account registration system. So you can request free access to that Chimeras account by going to the Proteome Discover product page. So if you just go to Google, you type Thermo Proteome Discoverer, click on the link, and then at the very top of the page, there's a link that says free Chimeras demo. And if you click on that, then you fill in your information and we'll send you a free account, which will give you a certain number of hours of processing, which is enough for you to be able to evaluate.
Thank you, David. And another question that's coming in multiple times, when will PD 3.2 be released?
Yeah, so PD 3.2, we're feature complete. The final testing now, so it's soon. I think tentative to give a hard date that's going to be out for sure, but tentatively in mid-November is when we expect the Proteome Discoverer 3.2 release to be available to all of you.
Excellent. Next question. Are the protein peptide counts displayed in the Data Explorer FDR filtered?
Yeah, thanks for the question. So that's a question I think referring to the Ardia display of the Proteome Discover results. So the answer to that is yes. We will not put numbers in the Data Explorer associated with the PD result file if there is some sort of control of confidence. So we don't want to put results into the Data Explorer there for you to peruse what potentially the quality of the result is without having some sort of FDR control. So you don't get 30,000 proteins and some enormous uncontrolled number of PSMs and peptides, but instead we will only put high confidence results into the list there.
Very good. And David, we're getting so many questions coming in. Let's go with this next one. What will be the biggest advantages of PD 3.2 over 3.1 SP1?
Yeah, so the main improvements for PD 3.2 were in the slide that I talked about before. There's some major improvements to Chimeras. So Chimeras is significantly faster. It can run in memory larger data sets. So we've tested several hundred gigabyte size data sets where we do what we call quan and on all files to increase quan occupancy. New post-translation modifications, so we can do acetylation, ubiquitination, whether it's DDA or DIA, site localization for DIA data, and also I'd just mentioned for one of the other questions, in-Fisher scoring. So this prediction models that work for Chimeras also works for in-Fisher scoring. So now we can handle phosphopeptides, ubiquitination, acetylation, and so forth as well. And then we also have these improved interactions with Ardia. So the version, you need PD 3.2 to upload to Ardia 1.1 to see the list of proteins, peptides, PSMs, spectra in that Data Explorer application. And so we also have some new ways of presenting DI results in Proteome Discover with tables of precursors rather than peptide groups. So we've definitely, when you run DIA, you get it presented kind of like the way that all of you expect DIA data to be presented in the output. So that's a short summary of the improvements. There's still other kind of user interface improvements. And if you want to know more details, I can send you more information.
Thanks, David. And since we have such a variety of questions coming in, I'm going to give you a second to look at them real quick and let me know which ones we'd like to get to next.
Okay.
Okay, let's go with this next one. How do you guarantee the safety when using chimeras?
Yeah, so get this question a lot. First of all, if you have Chimeras on Ardia server, your data will never leave your organization because the Ardia server is on-prem and it's local. It's within your firewall, and so that will keep all your data in-house. Now, the version of Chimeras that's on the cloud, the information we send from PD in the node, we don't send raw files, so there's no sample information. What we send to the cloud is a reduced set of MSMS spectra. Of course, you have to send a FASTA file. There's also this thing called a Chimeras inclusion file, like Kevin mentioned, that is used for quant completeness. That goes up to the cloud. We do the search with all that in the cloud, and then we delete it immediately. So we are not interested in storing your data, but we only keep it in the instance on the cloud as long as we need it for the search. We do have a statement that we've created for these questions, and we can send that to you. That explains more detail about all the information that's sent back and forth. And we don't store your data at all. So once the Chimera search is done, it will download the results back to Proteome Discoverer, and that's the only place where that data will be located. So we do our best. So we just take the minimum information possible, and there's little to no sign of that search after the fact.
David, what if I want to use another software package for DIA?
Yeah, so of course, many of you are using other software platforms for doing DIA. It does not mean you still won't gain value from using the Ardia system. So the Ardia system itself, all these other features of Ardia, that includes data management, that includes instrument management, but also you can store your raw data, download using a tool called Ardia Platform Data Sync. You can actually upload the results from third-party tools back into Ardia for safekeeping, so you have that large hard drive. But also, even if you're going to do the full analysis with another software package, there's still value to running Chimeras on the fly, file by file. And you can see, again, in that Data Explorer application, so that's the application for file management in Ardia, you can see consistent results from one analysis to the other. So you may not create a multi-consensus report out of PD, but you would gain benefit from using Chimeras on the fly to at least measure your samples as they're being acquired.
David, I think we have time to maybe fit 2 more questions in, but I do want to encourage our audience for those questions that we are unable to get to, and additionally, those that are submitted during our on-demand period, they will be addressed by our speakers via the email address you provided at the time of registration. So, okay, let's dive into this one. Can a server tower house both Chimeras, Ardia, and PD together, or is it recommended to separate the two?
Yeah, thank you for that question. So the Ardia server actually does not run Proteome Discoverer on the server itself. So the Chimeras algorithm is on the Ardia server. The Ardia server has a special hardware setup to run a local Chimera service, but Proteome Discoverer is installed on a separate Windows PC. So the data flow that happens when you run the automation is Excalibur will acquire the data. It'll upload the file up to the Ardia server. And then if you associate that raw data file with an analysis method, it will trigger a job on the separate Proteome Discoverer PC. So there's always going to be a separate Proteome Discoverer data analysis PC that will be running almost all of the workflow in PD except for the Chimeras part. So then the Chimeras node in Proteome Discoverer for Ardia server will trigger a Chimeras run on the Ardia server, sends that data back to Proteome Discoverer again, and then Proteome server finishes the workflow. So there will always be two separate PCs. There'll be an Ardia server, and then there'll be a Proteome Discoverer processing PC.
Excellent. And while David's choosing our last question for the day, I do want to address the question that's come in multiple times. Is today's presentation downloadable or is this video recorded? Yes, it is. Today's presentation will be available for unlimited on-demand viewing following today's presentation, our live presentation, and Laborats will alert you via email when this broadcast is available for replay. So be watching your emails. Okay, and David, we have time for one last question.
Yeah, I got to choose one. There's so many.
Yeah,
to choose.
so many.
So,
Okay, let's go with this one. Can you use Ardia to store data from other instruments?
So the answer to that is Ardia is just an open file system. Well, it's not an open file system. It is secured by accounts and user management and so forth. But yeah, there's generally no limitations to what kind of files that you can store on that system. So you can use results from other software packages, other instrumentation, open formats. There's one thing that you need to do when you're administering your server is that there's different file types that are accepted by the Ardia system, and only those file types can be uploaded. So you can go to the data management system and you can add a new file type that's supported by Ardia, and then you can upload into Ardia whatever you want. So Ardia can be used just as a general kind of storage server, but if you're using Proteome Discoverer results, if you have Excalibur raw files, it'll treat those a little bit more special than the other ones, with the annotated protein peptide IDs. If you have a raw file, which we call an injection, you can open up in an application called Data Viewer. So there's more things you can do with our data formats, but effectively you can store any kind of data you want on the Ardia server.
David, thank you. And before we close out, do you have any final comments for our audience?
Oh, thank you. Sorry. So sorry I couldn't get to all the questions. So I was looking through the rest of the questions and wish we had time to answer them all. So thank you for sticking it out through the webinar, and thank you for all the great questions. And for those of you who have not gotten their questions answered yet, we will follow up very quickly with a direct email. And thanks again for your attention.
And David, thank you. And thank you to Kev, and also for your time today, for your important research, and for this great Q&A session. So many great questions. I do want to thank Laborats and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I want to thank our audience too for joining us. Yes, we had lots of questions that we were able to answer and many that we were unable to answer. We do have your email and they will be answered offline. So thanks for participating. And today's webcast, again, one more time, is being recorded and it will be available for on-demand viewing. So just watch for that email and we'll let you know when it's available. And we encourage you to share that email with your colleagues and your friends who may have missed today's live event. I do want to thank you again for joining us. Until next time, take care, everyone. Have a great day.
Bye-bye.
Bye-bye.